professor at the Institute of Advanced Studies of the Strasbourg University, where she holds chair in therapeutic immunology. She's also emeritus professor from she's also emeritus research director of Drug Discovery Center for Cancer and Inflammation. It's a laboratory of excellence. She received several national and international awards, including CNRS Silver Medal, CNRS Innovation Award, and she is a fellow of the European Academy of Sciences and also member of Academia Europea. She is a co-founder of Neosystem, today it's called Polypeptide France, and also co-founder of Immune Pharma France. Professor Muller is a very eminent scientist in the field and has filed over 30 patents and published more than 400 papers and reviews in peer-reviewed journal. And I would like to thank CEFIPRA for organizing this lecture series, and we are all looking forward to hear your presentation. Yes, Professor Muller. Thank you. Thanks for coming, and we are looking forward. Yeah.
Thank you very much for your introduction, your kind introduction, and for this presentation today. So it's interesting because I'm also working in the International School of Biotechnology, Strasbourg, and so where we have many students and collaborators, of course. And so we will see that we have some similar lines of interest. So in fact, I worked with for a long time with autoimmune diseases, and perhaps for those here who don't know exactly what is autoimmunity, I prepared this slide to say that sometimes the immune system recognize by mistake our own components, and therefore they attack our own cells and tissue like foreigners. So this is seen by the appearance of autoantibodies and cells, lymphocytes. They react with our own component, and they are called autoreactive T and B lymphocytes. So in fact, autoimmunity exists in all of us at a very low background and without any damages, but sometimes for different reasons we can develop autoimmune diseases. And in this case, this is the progression from the benign autoimmunity we have everybody to pathogenic autoimmunity. And it is determined by several factors like the genetic, but not only, but also a trigger. And this trigger can be stress, environmental factors like pollution or different chemicals. And also some, as you will see, some hormones dysregulation. So we estimate, but perhaps the figures are understated. We estimate that 5 to 8% of the general population in the world has an autoimmune disease. So it's an enormous number of patients. So personally, I work on lupus, systemic lupus erythematosus, and about 5 million people have this disease in the world. And as you see here with the figure, most of the patients are women. And in fact, it's due, as I said, to the regime is due to several genes. And this panel of genes is not the same in the different populations in the world. It would be too simple. And due to environmental factors and also hormones, as you can observe with this disbalance towards women. And at the level of the organs, you see there are many defects. Skin, joints, kidney, brain, heart, lungs, a lot of things. And you can have all these defects. And this is a typical rash you can observe. But okay, it's not very nice for a woman to have this rash, but this is not the worst. The worst is really brain disease and heart disease and vascular disease. And there is also this form of neuropsychiatric form. And in this case, 10-fold increased mortality rate is reported. And there is also a form of neonatal lupus, but in fact the baby is not affected. This is due to the passage of autoantibody from the mother across the barrier, and these antibodies will bind to the fetal heart and create damages in the fetus or in the baby. So we have to introduce, to predict that, and to introduce a pacemaker to the baby, but still we have at least 10% mortality at birth. So, and for all that, we have no cure, no cure. So what you have, of course, we have treatments, but they are personalized, of course, because we don't treat similarly a person with brain disease or kidney disease, but they are palliative. And this immunosuppressive drug we use, particularly cyclophosphamide, introduce an immunosuppression, a global immunosuppression, not specific immunosuppression, and with deleterious side effects. So I wouldn't say that it's not good, because in the past, before cortisone, for example, half of the people died 1 year after the diagnosis. So today, the figures, as you can see here, are not the same. We improve enormously, but the quality of life of patients is very poor. It's very poor, and after a while, the people become non-responders to the drug we give, or we have to stop due to the deleterious effect. So the mortality is mainly due today to renal disease, cardiovascular disease, and infection. Because when we give high doses of corticosteroid and immunosuppressive drugs, you diminish all the resistance to pathogen, and people die from infection. It's true drama. So I will show you some data we have, not with an immunosuppressor, but with an immunomodulator. It's completely different. And the name we use for the drug development in the patients is Lupus. And so it is among the few molecules today in phase 3 clinical trials. There are many tested today, more than 70, but not at the level of the clinical trial phase 3. So just a few words. In the lupus, the active molecule is a peptide. The peptide is called P140. So P140 was discovered completely by chance. Completely by chance. It was not predicted. It was not the object of the aim of our study. It's often like that, you know. So in fact, at that time, we were screening a battery of overlapping peptides covering a protein which is called U170K, which is present in the spliceosome. The spliceosome is composed of several proteins, U1A, U1C, U170KB, and a protein that are similar to different particles. And there is also a small RNA contained in this particle. So we screen the cells from mice. They develop the lupus disease. They are MLR LPR mice. And we found that one peptide was able to induce proliferation of these CD40 cells. After we asked the question, why this peptide? What is the particularity? So at that time, we were quite concerned with the fact that antigen can become auto-antigen after modification by cross-translational modification. So we introduced phosphorylation, acetylation. By synthesis, you can do what you want. And in fact, we discovered that when in the sequence is this peptide. The sequence here, we modify this lysine here by a phosphorus, this here in the thing, this serine 140 by a phosphoserine will increase the activity. If we acetylate the peptide at this two positions here on lysine, we have no reactivity whatsoever. So, some years later, we discovered that T cells from patients were also able to recognize the same peptide. However, in contrast to what we have with mice, we have no proliferation in this case with patient cells. We had a blockage of the proliferation, no secretion of certain cytokines, in particular pro-inflammatory cytokines, but secretion of IIT, an immunoregulatory cytokine. So we continue with this particular peptide, and we showed that this peptide injected to lupus mice was able to delay the mortality and to establish a number of clinical signs. Dermatitis, less proteinuria, showing that we decrease the dysfunction of kidney, less destruction of renal glomeruli, less vascularitis in kidney, and also decreased levels of antibodies to double-stranded DNA, which is a marker of the lupus. So, this peptide was, of course, the sequence was patented just in time before the defense of thesis in the lab. And then we created a small company to develop this molecule. And the name of this company is Immunologist. And then all the different steps of the clinical trial progression was done. Phase 1, phase 2A, phase 2B, etc. And I like to show all these dates to illustrate that in an academic world, we are not Merck or big companies able to develop drugs with 1,000 or 2,000 people for each product. It's very long. It's a very long process, and we have to be courageous and really motivated. Anyway, so we had some success, really impressive success in the phase 2B, and then we had a first phase 3 organized in different countries, and in particular in Europe, in Mauritius, and also in the States. In fact, we had very good results in Europe and in Mauritius, where, as you know, the great part of the population is of Indian origin. So the figures were quite impressive in terms of responder to the product versus the placebo. However, it's like that. Very disappointing. In the US, we had no difference whatsoever between the group of treated patients and the placebo. So in the US, it was really negative. While in Europe and in Mauritius, with really different population, we had a good success. So today we are repeating, just in the US, we are repeating a new phase 3 with a US company called AIIMS, and this is running presently. And we also start, and you will see why later, we also start a new clinical trial out of the lupus context with CIDP, which is neuropathy in the peripheral nervous system. Okay, so just for you to know a little bit more about this peptide, as I said, we know the sequence, of course. It contains the phosphoserine at position 140, and this position is effectively phosphorylated in relation with apoptosis. So this is really one of the first change in this protein when the program, the apoptotic program starts. The non-phosphorylated peptide displays no protective activity, and the peptide alone is non-immunogenic, something very important when you consider therapy with immunoglobulin, for example, antibodies to something cytokine. We, of course, the PK data are very good, and we are very lucky that the peptide is highly soluble and quite stable. So it's a dream for somebody who wants to develop a drug. So we know we have the NMR structure of the phosphorylated and the non-phosphorylated peptide, the phosphorylation +1, and we also discovered several years later, that the peptide is present, in fact, in a loop here, just at the surface of the big complex. And this structure, this X-ray structure, was published at least 10 years later compared to what we discovered and what we do. So we know today how the peptide distributes within mice. So after intravenous injection, the peptide will accumulate in the spleen, and in a particular zone where there are a lot of macrophages and lymphocytes. And this is important because there are antigen-presenting cells. We also know, thanks to another PhD student, that the peptide can enter MLPR, so the lupus B cells, via the class-ring pathway, and will reach the early endosome, late endosome, and finally lysosomes. So we did that with different markers and different inhibitors to prove the pathway of the peptide. Another PhD student, very active, as you can see with the publication, identified that the receptor of this peptide is an HR protein called HSPA8, which has a particularity to be present at the cell surface all the time, in contrast to general HSP proteins, which are expressed only when there is a stress. Here it's constitutively expressed at the cell surface, and even more in case of inflammation. So we know where the peptide attach on the protein. So this has been studied in the literature. So, and what we discovered that when the peptide interacts with HSPA8, it hampers its chaperone function. It's very important, and those dependent. And P140 also reduces the interaction of the HSPA8 and co-ligand at the cell surface. So what could be the consequence of destroying this complex at the cell surface? The consequence, if it's true, that at the cell surface, MHC protein should be less stable because they are not any longer protected by HSPA8. And if they are destabilized, they will present less peptide or different peptide at the T cell receptor at the surface of T lymphocytes. It's true, and it is true, because we observe that here normal mice, MLPR lupus mice, and when they get older, we have a hover expression of MHC protein at the cell surface. And then when you treat, you have a decrease. And you have the same thing in the case of lupus patient. When we treat ex vivo, the cell completion, we have a decrease of this overexpression. And if you use a stronger peptide, you have no effect whatsoever. So here we destabilize this green receptor. Okay? What is the consequence of that? The consequence of that, downstream, is that this T cell should react less well, since it receives less signal. And it is exactly what we had after P1 treatment here in the range towards different epitopes, autoepitopes. So we wanted to say, to show whether then we have an effect at the level of B cells, still downstream. And effectively, what we observe is that both in mice and in patients, we have a decrease of plasma cells and immunoglobulin production. This was done in different papers, and this was really repeat and key in this function. What we observe also, and this is very important, is that the peptide doesn't react with the PCR at the cell surface of B cells. It has to act via the APC, all this process, not directly here. This was published also with McGill in Montreal. So, everything is very good. But then we say, well, perhaps we will have an effect in all the immune response. So what we did was to treat a series of mice with peptide, and we kept the other group untreated. And we infected the two groups with flu viruses via the intranasal route. And we observed no difference at all. So they recovered perfectly well from flu infection and were able to mount an immune response to CD4 T cells, epitopes, typical of the flu virus. So we can really conclude that the peptide is not an immunosuppressor, it is an immunomodulator. So here in brief, I recapitulate the whole process. So the peptide enters the class-3 endocytosis pathway, reach lysosome where it interacts with HSP8. HSP8 is also present at the cell surface, also here within the lysosome. This destabilizes AHC2 molecules, and all the process is blocked. So we have less activation of CD4 lymphocytes, and all the process downstream is decreased. So another view, the same thing. This is untreated situation. It is at the end of the day production of the auto-antibodies that bind to different diseases. When we treat, we abolish quite upstream the reaction. We have no antibody access. And recently, we developed another mouse, and a pure mouse, but with a defect of T cells. And we have exactly the same thing, which shown like completely other way that it is really the fact that we block the stimulation of this CD4 T cell that abolish all the process. So you will perhaps ask me why we have a separation, distinction between the self and the non-self. And in fact, we discovered relatively recently, and with the help of Anna Maria Cuero in New York, that the peptide blocks the process of autophagy. I think you know what is autophagy. It's a process that at the end of the day conduct the cleavage of the protein we are producing in high amount, or they are deformed, or they are abnormally they are defective in small peptides, and these peptides are presented by MHC molecules, or they are recycled to use the amino acid proteins. And we know that the peptide acts exactly here. It blocks this process. Again, this process is elevated, abnormally elevated in the because it's very useful. So we determined that with different markers that after treatment with P140, we slow down the excessive autophagic flux in molecular in B cells from molecular mice. We also demonstrated the effect of markers of chaperone-mediated autophagy or CMA, both in vitro, this was published here in autophagy, and in vivo, more recently in another paper. However, the peptide has no effect whatsoever on mitophagy. So no effect on mitochondria, only effect of lysosome-dependent autophagic processes. So it's by chance, it's difficult to develop a molecule like that, but it was just a discovery. So we asked the question, okay, it's fine. Now we have something and we have clinical trial, and I hope at the end of the day we will big success in lupus, but perhaps we could use the same molecule for other contexts, other pathology, where you have something abnormal at the level of autophagy. And so we started a series of experiments with experimental animal models. First, we started slowly, with neuropsychiatric lupus, and then with Sjögren's syndrome, where the salivary glands here are very active excessively. In CIDP, I talked about that before. Even with asthma, which is also a hyper-inflammation status, but nothing to do with the community, as far as I know. IBDs, we just published the data this year. Gum disease, inflammation of gum, so like that. And we have a paper submitted now. So I like to present this because it was done with the Indian PhD student, and she did very well working with us in Strasbourg, and we published 2 very good papers. And she developed the concept of the neuronal polarities in our lab, induced by ESS and TLDS. And she also used a transgenic model we obtained from the US. And as it appears, we have a lot of data. But just to illustrate, when we treat with a peptide, we have a very good survival, in fact 100%, while with a stronger peptide, we have really low survival. The body weight was recovered after treatment, and the length of the intestine was again recovered normally. We had in another model less blood into the stool, and of course the clinical indices for activity was good. It was 3 models. So here very rapidly, the mouse model of Sjögren's syndrome. Here we just illustrate that you can read the paper. There is less infiltration of inflammatory cells in the salivary glands. And here in the rat model of CIDP, we have also quite good effect on the IL-17 cells in the sciatic nerves, and less we treat change with macrophages. So all that is inflammation. Recently, in the gum disease, we had also all these the same similar results. But why I illustrate this is because we observe that not only we abolish the hyperinflammation of the gum, but also we protected the bones. And this is something very important because it's alter the or diminish the inflammation of the bone, but then the people have really harm in the bone, and so we preserve the quality of the bone. So this is also a plus we discovered recently. And here in asthma, we had also in the lung some very impressive results. So all that is published, and what I would like to conclude to tell you is that in all these models of inflammation, mainly chronic inflammation, we have of course an amelioration of the clinical markers, as I show you, preservation of the bone, amelioration of the excretion of pro-inflammatory cytokine directly in the target tissue. The biological markers are better expressed, and the autophagy was also regulated. So you will tell me that there are in the catalogs quite a lot of molecules able to modulate autophagy, inhibitors, activators. But the premise of our peptide is that the P140 block one particular step, not the other, and not other pathway of death, no apoptosis, and no effect whatsoever on something else. So this peptide by chain is extremely specific of one process, and this process is altered in several autoimmune disease or inflammatory. So, as I said here, it's a mechanism-driven therapeutic mode of intervention, and this is important. It's not against everything. Also the fact that the peptide targets the autophagy process, this means that it will only affect the autoreactivity and not the whole immune system. This is also important, and it explains why all the hundreds of patients that who have been injected today with the Lupus Erythematosus, P140 peptide, has no infection, no secondary effect whatsoever. In all our clinical trials, never whatsoever we had some secondary effect. And this is really unique compared to the other drugs. Another point, it's the dose we injected. You are biotechnologists, you know, you can estimate the figure here. We inject 200 micrograms of peptide in the whole body, not per kilo, in the whole body. It's nothing, it's really few. And of course, this is also a factor that explains the low toxicity of the peptide. And we inject the peptide once a month. Perhaps we could delay the number of injections, we don't know. So this avoids secondary effect, and something even more, the peptide is non-immunogenic. And this contrasts with the immunoglobulin we can give to patient today. After a while, the patient develop antibodies against the antibody, and they become normal. And of course, it's a chemical product, so you can produce large amount, really well controlled in terms of purity and quality of product. So today, what we are trying to do is to change the mode of administration. Today, we treat the patient by the sub-Q way, and mice in general by the IV or IP way. So my purpose is to now to try to define a non-invasive administration, oral, nasal, transcutaneous, and this needs to associate the peptide to nanocapsule or nano object to really present the target through the mucosa. We also try to develop some system to distinguish the responder versus the non-responder. It's very important because not only to include the good patient in the clinical trials, but also later on in the true life to avoid injecting patients who are non-responder for any reason. So to really specify the population to be injected. So I finish here, and I thank all my collaborators. As you see, there are quite a lot, and they were engaged with me for all the story because I speak that this is their work, and this is important to have the name of the several PhD students, postdoc, engineers, technicians with me, and this is our team today. It's a small team, but we are really motivated, and so it's, we hope we will, at the front of you, continue to identify all the process and accompany the companies who develop the peptide at the level of clinical trial. And I'd like to thank you for your invitation here, because it's really a pleasure to be here, and of course CEFIPRA for allowing this travel and my tour to be possible, and CNRS as well. Thank you very much.
Before opening, just have one quick basic question regarding the responders and non-responders in your initial trial. Since you mentioned that lupus patients present differently, every individual is different, were you able to do any type of correlation with that regarding your responders versus non-responders? And also, does ethnicity play any role, because you had a massive difference between the European population and the US population?
Yeah. I'm a scientist, so I don't have access to the material from the clinical trial. And it's very difficult to develop a clinical trial because you have to define before starting what you will do. For example, if you want to collect the cell from patient, you have to say before. So it's no way to say, oh, the results are what they are. I would like to have access to cells. It's too late. So I discovered that. Now I'm more cautious, This is the first point. The second point is that the phase 2B was done mostly in South America and Spain. So 70% of people were of Spanish origin. Then in Europe, we have our diversity, but it's what it is. But in Mauritius, it's still different. So I don't think that it's the cause of the discrepancy, I would say, is due to a genetic factor. Our feeling is that the population that has been selected for us for this clinical trial in the US was different. Of course, we had guidelines to say we want to include this kind of patient, but we doubt about that it was well done. Because it's really not normal to have 0 in one is big, but one country or one region of the world, and okay, everywhere else. So we think that the selection of patients was something wrong. So it's why we repeat, but in the meantime we lose time and money.
Thank you for this great presentation, excellent piece of work. So I don't know whether you mentioned in the introduction. So I wanted to know whether the peptide P140 is it designed to be resistant to protease activity? Was the sequence modified so that no MMPs can recognize it?
So the peptide is of course come from a real protein, but we synthesize. So the peptide doesn't exist in vivo, never. We know, of course, some proteases are able to cleave, but we never identify the peptide, for example, in the blood or somewhere. So it's
okay, and because you have changed some of the functional groups, you have phosphorylated the serine group and
so
all.
this phosphorylation exists in vivo. So this is the question I asked to a postdoc who came from the medicine in my lab. I said, perhaps never exists. And we discovered that yes, it exists, and this is the first change in this protein when the cell progress in apoptosis.
Thank you.
I have a related question. So I think in slide 10 we saw this peptide is a part of a protein.
Yes.
So this on this stone, that is it possible that in US community this particular protein is overexpressed?
It's a very good question. I don't know. Some say also the protein is involved in the spliceosome, and I don't know whether autoantibody from patients directed or cells against this peptide have an effect on this process. I don't know.
And also said the serine has to be phosphorylated.
Yeah.
And so, but you are injecting the peptide, and this has to go, and then serine has to get phosphorylated to have the action.
Yeah, without the phosphorylation, there is no effect.
So what you are injecting is the peptide sequence.
Yes.
And that has to get phosphorylated in vivo, or?
No, I inject the phosphorylated peptide, the synthetic peptide. Yes. And but to prompt your question, within, because a reviewer asked this one, within the lysosome, the peptide is very stable and remains at least 24 hours with the phosphorylation and intact. While if we inject the non-phosphorylated peptide, it is destroyed nearly immediately, in 1 minute.
Thanks.
It was a fantastic talk. I really enjoyed. One is just in continuation of Dr. Nadia's question. Like, if you look at, since you said genetic factor may not be the problem to see the difference between the responder and non-responders. But if you look at the same analogy for the cancer treatment, whatever the drug is developed against Caucasian population is not working for other population. But the major difference what they see is the genetics, right? Maybe it's a good to look at the genetic difference between these two population, whatever they use for the clinical trials. So you may be able to see something important. The second question, my question is, because you said this peptide 140, it blocks the fusion between autophagosome and lysosome. And we do know that there are a couple of inhibitors that are available like faplamycin and chloroquine.
Yeah, whether this is acting at different sites, for example.
Different site, basically based on the picture, it's blocked the fusion between autophagosome and lysosome. And chloroquine, faplamycin doing the same thing.
Yes.
Suppose can you see the same effect?
No.
Then how do we say that blocking the fusion?
The point is that chloroquine is active in lupus patients. Yes.
Yeah.
Yes.
That's what.
The point is that because it has other targets, not only here, it has secondary effects. So at the end of the day, some patients cannot receive the proxy. We have to stop it, even it's effective. We have to stop it. We can't continue with this. You know? And it's why I insist here, it's because the peptide really at the one point and not everywhere in different metabolic processes that it has advantages over the other molecule.
Okay, thank you.
And your first question was interesting, Dr.
No, the responders versus non-responders in your phase 3 clinical trials.
What I think is perhaps we will distinguish people by the receptors at the cell surface. So this has perhaps something to do with genetic.
That's what I'm trying to.
Until we don't know. We know that HSK8 is important, but perhaps something else. And this something else, I don't know what it is.
No, for example, if you look at pediatric neuroblastoma, if you look at in years, you see 85 to 95% success for the same. If you give the same medicine, same drug to other population, hardly you see 10%.
So it's why now we are trying to identify the protein at the cell surface, and when we know protein question, perhaps we will discover that this protein is expressed in certain patients and not in others. And of course, the other will not be responded. I don't know. It's a proteomic.
Thank you.
In conjunction with this question, is there anything to do with the HIV? I don't know. As long as European.
It's a large panel, screening, large screening, mice and human, no discrimination. So this peptide can have effect on any
Processing.
Yeah.
Even with the presentation and the hepatitis.
Yeah, exactly.
So another question was in continuation of the here. I remember showed it's a TB strengthened immune responses.
No. At the very beginning, when we published our first results, we thought that with this altered peptide, with the population, perhaps it could act as an altered peptide. But after a while, I realized that, okay, perhaps, but it's not possible that it acts only like that. Otherwise, we will only decrease or change or drivers the T cells that recognize the parent peptide. In lupus, how many the diversity of proteins are very large. So if you decrease one population, it's not enough to explain the success. So it's why, perhaps, yes, it is important, but it's by far not enough.
So I don't work in this field, so my question might be pretty naive. I'm sorry. You said that many of the autoimmune diseases, there are environmental triggers, and earlier you also said that things like chronic stress could be a trigger too. And then there are genetic factors. And we see pretty much in individuals that the autoimmune disorders develop rather late in life, like type 2 diabetes or even cancer. So I just wondered whether it's not just a matter of genetics, because genetic factors are always there, environmental triggers are always there, but something in between, that maybe there are epigenetic changes which occur, which might then lead to certain kind of triggers, and these kind of changes occur rather later in life.
Yeah, it's your question is in fact very good. In fact, the people are autoimmune when at birth. They have the gene predisposing genes, but then you need a trigger. And the trigger, again, will induce a number of effects, slowly and slowly. In lupus, in general, the diagnosis is around 16, 17, when in addition you have the change in the hormone pathway. In other diseases, perhaps later, can be later. What we don't know today is why in a family, for example, some patients have lupus, other rheumatoid arthritis, other scleroderma, whatever. We don't know why, what differs. It's relatively rare that in a family you have all the members, women members, with lupus. And we don't know why. So, and then it's slowly, you have a degradation, and then you have the symptoms. So it's why it's no way to treat the patient with the lupus drug, for example. Because you treat, you block this power of events, and then, okay, patients are recovered. After a while, because we didn't change the genetic, so after a while, the bad cells, the deleterious cells and antibodies will reappear, and then you treat again, and then you treat again, like that. So there is a space, a delay between two injections, just to block what can come again. You know, it's really for chronic disease. It's really. So. We have preliminary results with glioblastoma, but still in vitro. So, but I think that in different conditions where you have defect or alteration of the autophagic process, you can infect. Thank you for your question.
Excuse me. One of the question that one of the topic that you discussed about was HSP protein, the HSP8 that you were discussed about. Are there any other proteins also which can stabilize the MSC class 2, which will rise up after the blockage?
It's essentially HSP8. So the old name of this protein is perhaps HSC17. It has 7 names. This is really the job of this protein to protect, to chaperone the eligibility.
And if the B lymphocytes are not getting suppressed by any way, so there are any other ways by which, apart from the CD40 ligand, apart from that interaction, the other way also for the B lymphocytes to get activated, if there is any?
I'm not sure I understand your question perfectly. Can you repeat?
The CD40 ligand interaction between the T helper cells and the B lymphocytes.
Yes.
So you say that the B lymphocytes are not getting anyway inactivated.
But they are inactivated because they don't receive a signal from the T cell, you know.
Okay.
So everything around is disturbed, but not the good cells, only the auto-reactive CD40 cells.
Responder individuals, responders, okay. So, but like they were very uniform, or there is specific like nephrotic patients or inflammatory patients? Are there any variation in the responder?
You know what we don't know today, and because we don't have the complete reception, we don't know why certain cells will have HSP8 at the surface and perhaps the other complex, and some other cells in the same individual have no reception. But it's clear that the peptide, remember, we inject 200 micrograms, really a long dose. So if we consider that if the molecule peptide will enter all cells of our body, it never works, never. So it clearly targets certain cells and not other cells. And in one organ, for example, the spleen, some cells have the complex surface and some others not. So how the peptide knows the cell, we have really to discover the complete receptor within the responder cell.
So in the introduction you said when you had a true acetylation in the adjacent ligand, the activity reduced.
No activity whatsoever.
No activity.
No activity.
In the phosphorylation.
And here we have model of the peptide effect. So you have the NMR structure. And so we this acetylation really destroys the structure,
The conformation.
change the structure. And you know, at the very beginning, we had this screening with synthetic peptide, and we made, I don't know, 50 or 100 analogs of the peptide. And more or less, we cannot change one single residue in this peptide. The first peptide was a good one. We can only play, unfortunately, with the N terminus. So at the N terminus, you can add biotin, fluorescent reagent, everything. Also put on beads or whatever. But the rest of the peptide cannot be changed.
Yeah, I was thinking, addition of the phosphate also negative charge and acetylation on the figure. So probably that.
But when we model, it's really changed the structure. And probably the interaction with the receptor.
Any other questions from students?
Don't be afraid.
No, they ask the best questions, but you have to give them some time.
Anyway, you have email address, and if you have any question, you can feel free to ask.
Okay, if there are no questions, we thank Prof. Muller for coming. It was absolute pleasure and it was wonderful.
Thank you so much for this exceptional day. I will always remember, always. Long live India and France friendship.